Thank you very much. I'm really pleased to be here. Thanks for the invitation. For those of you taking a look at the title, Eat, Drink, and Be Scary, I decided in reviewing for the presentation I don't have enough time to cover all of those, so there'll be no scary parts, but there'll be some eat and drink parts, at least in the background. I'll be talking about how we're using genomics and systems biology to analyze some specific aspects of social behavior, covering two topics today. First, the evolution of eusociality, and then secondly, a more mechanistic perspective where we look at trying to understand how changes in gene expression are regulating changes in social behavior. First, let's begin with a little bit of a background. I think we can all agree that social evolution is a really potent force, and it has given rise to some of the more spectacular traits on the planet: language, agriculture, and, for better or for worse, warfare. You're probably familiar with the manifestations of those traits in our own society, so let me just quickly touch upon them in insect societies. In the honeybees, we have a symbolic language, the dance language, the only species that communicates with a symbolic language, except for us, perhaps. For agriculture, we have the famous leafcutter ants that have been studied extensively over the years, and recently their genome has been sequenced, and beautiful aspects of genomic interactions between symbiotic species within the leafcutter group have been elucidated by Cameron Curry. I think he'll be speaking here during this conference. And then finally, an example of warfare. We have the harvester ants in the desert Southwest, where different colonies engage in tournament behavior. So language, agriculture, and warfare all exhibited by the insect societies. So when we stop and think about this, we see that these kinds of traits have a very puzzling phylogenetic pattern. They exist in the insect societies and in the human lineage. And the last common ancestor of these two groups lived some 600 million years ago. See the picture here below is the depiction. It is thought to be a very primitive marine flatworm with a very rudimentary nervous system, no social life to speak of, raising some profound questions about the role of conservation and convergence in the evolution of social life. And to that traditional array, convergence and conservation, we now can include a newer concept, which is the evolution of toolkits, genetic toolkits, that are repeatedly tapped by evolution. And that's the kind of concept that we're working with and that you'll see sort of embedded in the talk today. So, let's first define eusociality. That's what the insect societies have to offer with respect to social behavior. It is the most extreme form of social life known. It is characterized by three traits: an overlap of adult generations, and then the key trait, the tremendous skew in reproduction. Some individuals reproduce and some don't. In most insect societies, we refer to this as the queen who reproduces, or multiple queens, and then the workers who don't. And then the workers engage in cooperative brood care. These are the three defining traits of eusociality. What I'll be telling you today are some stories that give us some insights that feeding, nutrition, metabolism, and reward are implicated in the evolution and functioning of insect societies. And I'll be emphasizing today primarily nutrition and metabolism. So let's begin with some evolutionary analyses, first of all. An idea that was developed a number of years ago by Mary Jane West Eberhard posited that when you take a look at maternal behavior, you can decompose it into two parts: reproductive behavior and care behavior, the actual care of the offspring. And what Mary Jane West Eberhard observed is that in the insect societies, this traditional coupling of reproduction and care is uncoupled. So the queens engage in reproductive behavior. They're the egg-laying machines. By the way, they don't control or regulate the society. They don't tell the workers what to do. Their job is to lay eggs. And then the care portion of the original maternal behavior package has been separated and has now been taken on by a different group of individuals, the workers. So Mary Jane West Eberhard posited that part of eusocial evolution was the uncoupling of these two components of maternal behavior. Lingspear and Wade, a number of years later, put this into molecular terms, talking about the concept of heterochrony and the separation of packages of genes, programs of genes that are expressed not only at different times, which is the traditional concept of heterochrony, but taken to such an extreme that they are expressed in different individuals. So this set up a very nice test, which is that there should be differential expression of genes associated with care behavior and reproductive behavior. We set out to test this idea, taking advantage of primitively eusocial insects, such as the picture that you see here, a paper wasp. Primitive eusocial insects they have crossed the eusocial threshold, they have the three components, but their societies are small, relatively rudimentary, and most importantly, very flexible at the initial founding stage. So a foundress, a single individual who founds the nest, at the beginning, she does everything. She's involved in reproduction and care. Then, when she lays her first eggs and they're successful and they emerge as the first generation of adult workers, she kicks back and becomes a queen for the rest of her life, and the workers take over. So we have these three different groups, foundresses, queens, and workers, with the foundresses showing great flexibility, sometimes looking more worker-like as they're in their single mom phase, and then sometimes looking more queen-like in the way we think of more traditionally with social insects. So we wanted to test the ideas that there's differential expression. But this species, the paper wasp, had never before been studied from a genomic perspective, so we first needed to sort of plant the seedlings, if you will, till the vineyard, get some resources, and be able to have the wherewithal to do this. So we used next-generation transcriptome sequencing, created a database of ESTs, and then in the first study, we selected genes from our studies of honeybee gene expression, differential gene expression between individuals showing care behavior versus those that are showing foraging behavior. So we had a set of hand-picked genes to be able to test the hypothesis. We started this off with the paper wasp, Polistes metricus. We've now been extending this to other species. First, a bumblebee, Bombus terrestris, also a primitive eusocial species. And what we wanted to do was look at the differences in gene expression between these different groups, between queens, foundresses, and workers especially. There's another group there, gynes, that we've used for a different purpose, which I won't be talking about today. But so let's focus on those three groups. And the basic hypothesis is that if the foundresses indeed are showing more worker-like behavior, we would expect their patterns of brain gene expression for these hand-picked genes already implicated in care to show a more worker-like pattern. Here you see two multivariate analyses that analyzed the results of the 32 genes quantified by qPCR, and you can see that in both analyses that's exactly the case. The foundresses and the workers more resemble each other in terms of brain gene expression than the queen. We then moved from qPCR and a hand-picked set of genes to creating a microarray for the paper wasp. And when you do that and just survey all the genes that are expressed in the brain based on our transcriptome analysis, our EST set, and we had about 3,000 genes, you see a different pattern. You see that the differentiation between the foundresses and the workers or the similarity between the foundresses and the workers that we saw with the hand-picked genes was not seen when we looked at all of the genes, suggesting that when we focus on these care-related genes, we get the pattern, but not across the transcriptome in general, which makes perfect sense. These results now have been extended to a bumblebee, where we see the care gene story holding for bumblebees. And I show this in this slide by showing a significant overlap in the patterns of gene expression between the paper wasp and the bumblebee in terms of brain gene expression across these three groups of foundresses, workers, and queens. So we see unlinked gene expression with respect to these patterns in these different groups, but we've been talking about genes sort of in an anonymous way. We also would like to know what genes might be under selection, what particular traits have been shaped, have been acted upon to create insect societies. Now here we operate at a bit of a disadvantage compared to comparable studies with humans. With humans, the dominant paradigm is the big brain. When you think about human evolution, in many ways, you can reduce that very complex problem to what genes have been under selection to give rise to a big brain, in particular to the expansion of the cerebral cortex. With insect societies, the problem is a little more difficult. Even though the insect society has attained comparable levels of complexity, of sophistication, of cohesion, it has been done in a totally different way. In the human trajectory, it has been an investment in the individual. So we have an increase in the capability of the individuals. In the insect society, it has been done more by an increase in the dependence of the individuals upon the society. So instead of a me paradigm, we have a we paradigm. So we need to find genes that are related to creating interdependencies and create the idea that the workers are working together, and in fact their behavioral capabilities in an insect society, so eusocial behavioral capabilities of individuals, are no different or no better in most cases than their solitary contemporaries now, and we assume they're solitary ancestors. So we need to be thinking more broadly about what kinds of traits could make the workers more interdependent on each other. And that's complicated, and you'll see the problems of interpretation as I show you the data. So we looked for molecular signatures of selection. For this, we focused on a more cohesive set of species, the bees, a set of bees that span, now insects are old, so even a cohesive set, fairly phylogenetically resolved set, involves a fair amount of evolutionary distance. We primarily looked at bees that are known as the corbiculate bees, that collect pollen and store pollen, hold pollen on their hind legs. We looked at 3 different kinds of bees. Bees that are not eusocial, they're indicated in green. The bees that are primitively social, the blue, and the highly eusocial species. Highly eusocial species such as the honeybee, large colonies, tens of thousands of individuals, very defined roles. Some of the more spectacular insect societies are in that group, such as the leafcutter ants, and honeybees and the tournament ants that I showed you before. Primitively eusocial, smaller societies, more flexible behavioral roles such as paper wasps and bumblebees, and then solitary or non-eusocial species. The set we have, as you see, included 3 independent acquisitions of eusociality indicated by the diamonds on the tree. We created brain transcriptome data sets for all of these, and then aligned these painstakingly because the evolutionary distance was such that automated methods of alignment were not sufficient, and then using the traditional approach of PAML, looked for signatures of selection. Here are some of the results. Now, I emphasize there were multiple independent evolutions of eusociality, and they've converged upon a set of striking traits that define this lifestyle, but this then would be convergent evolution. So we did find some genes that have been under selection, showing more rapid evolution. There were 212 genes when you compare eusocial to all other species, non-eusocial. Also some genes showing rapid evolution in the non-eusocial species, as we might expect. They haven't exactly been sitting still and not doing anything over evolutionary time. Also differences on the right-hand side between the highly eusocial and the primitively eusocial species. What were some of these genes? Well, when we did a gene ontology analysis of the genes, we saw some dominant trends, and they're indicated for you here. I won't go through all of them. I'll just highlight a few. One in particular, metabolism, figures prominently. We saw a strong hit of genes, signature of selection of genes involved in metabolism for all the eusocial species, and also when we compare highly eusocial with primitively eusocial species. The metabolism story was particularly strong. All the genes that you see highlighted in color here, we're not going to go through all of them, obviously. This is a KEGG pathway for carbohydrate metabolism. All the genes highlighted in color are showing more rapid evolution in either the highly eusocial or the primitively eusocial species. And then, of course, so we're excited about this result. We have a strong hit of selection in a number of biological processes. And then now the work has to come to figure out, well, what does selection on metabolism have to do with eusociality? I could take some time and spin some stories about the fact that eusocial insects are able to air condition their hive and heat their hive, the fact that they forage all day and take many, many trips and show unique relationships with respect to metabolism. The truth is we don't have a clue yet, and so this is just the first step. First, we need to take these hits of selection and map the amino acid changes, find out what functional changes there might be, if at all, and then begin a process of asking how these changes in such a, quote, low-level biological process, metabolism, metabolism could be involved in these high-level changes in social life. So we're just at the beginning of this, but this approach, aided by genomic resources, is giving us finally a foothold. The evolution of eusociality has been studied for years from a theoretical perspective, identifying the evolutionary forces, but I think in order to really ground those analyses, we're going to need to know what genes have been acted upon, what biological processes actually have shaped the evolution of insect society, and this is a first step. Okay, the second and final part of the talk is then to look at this in a more mechanistic context and take a look at transcriptional regulation of social behavior. We've done a number, and this is now focusing on honeybees. We've done a number of microarray analyses. Now we're switching, like many other people, to RNA-seq to measure changes in gene expression. In our case, we're interested in behavior, so primarily, but not exclusively, the brain. And we find that changes in behavioral state are associated with massive changes in brain gene expression. So we want to understand this better from the point of view of transcriptional regulation. The dominant biological themes that we're exploring here, again, relate to hunger and nutrition. And what we're seeing is that it appears that hunger and reward pathways have been co-opted during social evolution. They're regulating changes in behavior that seem to largely be independent of hunger states, which are the canonical states that these pathways have been associated with. And I'll be particularly talking about the insulin signaling pathway. What's interesting in general is that some of the brain gene expression patterns associated with insulin signaling look like bees that are going out to forage are hungry. But we know they're not hungry because they eat before they leave. They tank up on food in the hive. They live in a civilized environment. They've got stores of food. They eat before they go out. They're not going out because they just need a meal because they're dying of hunger. So yet their pathways seem to be giving us the signal that they are. So just briefly, what I'm talking about here is embedded in their process of behavioral maturation. Honeybees spend the first 2 to 3 weeks working in the hive, and then they graduate and become foragers. Hive activities include nursing and working, building the structure, and then they become a forager, collecting nectar or pollen. We found that fat loss causes precocious foraging. Bees lose about half of the fat in their abdomen prior to becoming a forager. That's seen on the graph on the left. So the nurses are much fatter than are the foragers. The nurses are in pink. And then this loss in weight, this weight loss, this process is actually causal. When we experimentally cause bees to lose weight, to lose the fat, by treating them with TOFA, the graph on the bottom, which drops abdominal lipid, we get an early onset of foraging, which is the top right graph. On the y-axis, the proportion of bees foraging, and you see that the bees treated with TOFA show an early onset of foraging. We also see influence of hormone signaling associated with nutrition. 2 hormones, juvenile hormone connected to vitellogenin, a lipoprotein, a nutritional protein that's involved in storing nutrients and secreting them and sequestering them and then releasing them at appropriate times, and then insulin signaling. These just quickly are 2 experiments that establish causal relationships here. On the left, we treated bees with RNAi for a transcription factor, USP, involved in hormone signaling. We reasoned that if we decrease juvenile hormone signaling, we will then lower this trafficking in this pathway and delay the onset of foraging, and that's exactly what we saw. And then likewise, we decreased insulin signaling activity, in this case pharmacologically with the treatment of rapamycin, and we delayed the onset of foraging there. On the y-axis in both cases, the proportion of foragers, and then age on the x-axis, and you can see the treated line, pink, showing a delay in the onset of foraging. So this led us to then explore more carefully what's going on transcriptionally with respect to hormone signaling. We picked on this particular transcription factor, USP, because its involvement in juvenile hormone signaling. USP, we showed, collaborates together, collaboration with Minong Lee's lab at University of Kentucky. And what you see is that this was work done in Drosophila, where we could dissect this more carefully. USP collaborates with a number of other transcription factors and cofactors to regulate gene expression. So we think this is one of the ways that juvenile hormone is causing the massive changes in gene expression in the brain that it causes, which I did not show you. We've also looked at to try to find out what genes exactly is USP regulating. So to do this, we did chromatin immunoprecipitation, followed by microanalysis called ChIP-ChIP, with an antibody we developed for USP. And we found lots of putative binding sites for USP, suggesting almost 800 genes regulating. We have a pretty good overlap, 50% overlap with comparable studies done in other labs in Drosophila. Many transcription factors being regulated by USP based on these results, suggesting a hierarchical transcriptional model, starting with the hormone and then transcription factors that mediate it. Here are some of the genes highlighted for you that sort of are suggestive of some stories, suggestive of a connection to epigenetics, the histone modification, DNMT3 genes on the bottom of the table. Hormone signaling genes, this makes sense. This USP mediates hormone signaling. The predicted USP site, yes column, means that we've also bioinformatically looked for the cis-regulatory element, the sequence of the cis-regulatory element in the promoters of the genes implicated by the CHIP results. And in most cases, you can see all those yeses, in most cases, bioinformatic confirmation of this. We're now in the process of setting up a luciferase assay to validate some of the more interesting genes that are putative USP targets. We've also been looking at other regulatory elements in the promoter regions of these hormone-regulated genes. Here are two that I circle for you. ADF1 is one that we see regularly showing up in all of our sets of genes of socially regulated genes, suggesting it may be part of a social cis-regulatory code for social behavior. FTZF1 is related to juvenile hormone signaling based in studies in Drosophila, so that's pleasing that it comes up, and I'll show you another connection between that in a moment. And that's here. So in addition to the work on a particular transcription factor, USP, we've also taken a broad view. We've done a number of microarray studies. We've been able to then collaborate with Nathan Price, a systems biologist, to reconstruct a transcriptional regulatory network of the brain involving 853 Bs, 48 different behavioral states, and I just have time to give you just the beginnings of these results. Basically, the model is able to include 25% of the genes out of 10,000 on our array. It has a very high predictive value, a predictive average correlation of 0.87. That is, you can predict the expression of a gene by knowing the expression of a transcription factor. So it assumes direct linear connections between transcription factor and effector gene. So what we found is a sort of a mosaic pattern, a set of transcription factors that are globally regulated, 4 of them that seem to be involved in regulating 3 different behavioral states, behavior states within these large category behavioral states: foraging, maturation, and aggression. And then a set of transcription factors that are active only in foraging and not aggression and not maturation, or vice versa. So a kind of a mosaic pattern of regulation. Keeping with the hormone theme of this talk, we see that nutrition and hormone-related transcription factors are prominent in this brain transcriptional regulatory network. So I mentioned FTZF1 before, and so here we see it again. It's a JH-regulated nuclear hormone receptor. It's known to be regulated by USP. Here we see it regulating a very strong module of genes in the model, and it's predicted to regulate several different transcription factors that are also targets of USP. When we do the gene ontology analysis of the categories of genes that are expressed by, that are regulated by this transcription factor, some familiar names come up: vitellogenin signaling, lipid metabolism, neural development, and behavior. So in summary, what these results are pointing to give us intriguingly similar overall connecting results to the evolutionary analyses that I showed you before, leaving us with the working hypothesis that division of labor involves evolutionary novel relationships between nutrition and hormone signaling pathways. And that means then that feeding, nutrition, reward, and metabolic pathways appear to be major players in social evolution. And similarly, another conclusion we can draw is that social evolution has proceeded via changes in gene regulation as well as changes in coding sequence. Thank you very much.
You have questions?
Yeah, I'm sorry, I had to motor through it. Positive selection. Positive selection.
And in that case, did you have outgroups? Did you have a comparable sample among non-social insects?
Yes, we had several of the bees. I probably went through this too fast, but in our set of species, several were not social. And I could go over that in more detail with you later.
Other questions?
Well, in your introduction, you said that the worker bees weren't going out foraging because they were hungry, but you then went on to show that lipid content dropped dramatically in that transition from, let's say, nurturing to scavenging.
Mm-hmm.
So in a sense, you sort of contradicted yourself there, didn't you?
Well, they may have some of the physiology of hunger, but the observation is that they will ingest food prior to leaving the hive. So in terms of absolute and immediate nutrition, they've got what they need at the moment. But are they in a perennial sort of starved state? That's a very interesting point. And in fact, that it may be sort of underlying the evolution of eusociality, where the winners lay eggs and the losers go out and forage. So that's a good point. But the fact is they eat before they go out.
Other questions?
Have beekeepers bred honeybees to have different social behaviors or foraging characteristics that you could use in an experimental context?
Beekeepers haven't. There are some significant impediments to continuous breeding, but several scientists have over the years. And so, for example, Rob Page, the University of California at Davis originally, now at Arizona State, continued a program that Walter Rothenbuehler started to breed bees that collect large amounts of pollen, and then the counterpart, those that collect large amounts of nectar rather than pollen. And we've been collaborating on some of these meta-analyses, the bees that, so we look at brain gene expression profiles. High pollen hoarding bees look like, going back to the previous question, look like pollen-starved bees in terms of their brain gene expression. So there could be something interesting going on in terms of genetic assimilation, where you start with this immediate hunger state, this phenotypic plasticity, and then that was bred into bees that just want more pollen. So that's one example of a useful trait. And then, of course, African bees, highly aggressive. Those have not been bred to be highly aggressive, but we've taken advantage of those. And if I had a longer talk, I would have done the scary part on African bees.
Okay, thank you very much.